Well, it's my pleasure to introduce Roni Haas, who's a postdoc with Paul Boutros, and has been at UCLA for a year and a half, as I learned, and did a PhD in Israel on classical yeast genetics, experimental, and then subsequently bioinformatic work. And she is going to share her new project on cancer radioresistance. She also received a really prestigious Postdoc Fellowship Award in the summer from the Prostate Cancer Society. Welcome.
Thank you. Thank you very much for the introduction, and thank you for the opportunity to present our work here today. So I'm going to talk about our proteogenomic investigation to characterize radioresistance in prostate cancer. Let me see, maybe I can fix that. Schematic. I can't switch slides. Wait. Sorry. Is the person that fixed that before is still here, or? Okay, let me try. Maybe I'll stop sharing it again.
And reshare again.
Okay. How do I do that?
Right here. No, share.
Oh, okay.
And then pick up the
Share.
That's it.
But do you see the slides okay on Zoom, or it's?
You're not in slideshow, Roni.
Oh, I see. Huh. Oh, that's what he did? He shared the desktop? Okay, maybe I should try that. Sorry for that, guys. Stop share.
Find the desktop.
Oh, the desktop, okay. Let's try.
If you go in the presentation mode.
Okay.
Okay, that's good.
That looks good?
Yeah.
Okay, and I can move slides. Okay, looks good. So, okay, so what we're going to talk about. First, introduction for what is prostate cancer and why do we study radioresistance in prostate cancer. Then we'll see how we model radioresistance using cell lines and characterize radioresistance from several different angles, genomics, transcriptomics, and proteomics. And finally, how we took top targets and assessed their relevance using patient data. Let's start with some background about prostate cancer. First, it is a very common cancer type. 1 in 7 men will be diagnosed with prostate cancer. The mortality rates are around 25%, which is not very high compared to other cancer types. But the fact that prostate cancer is so common makes it the second most common cause of cancer death in men. And in prostate cancer, as also true for other cancer types, early detection is very critical to help improve patient outcome. And we are pretty good in early detection in prostate cancer, and many of that is thanks to the PSA test, prostate-specific antigen. It is a simple blood test and also not an expensive one that can detect prostate cancer in a very sensitive way. But on the other hand, it is also not a very specific test, so there are many false positives. So in order to diagnose prostate cancer, there is the use of several tools together. One of them is DRE. It is a physical examination for prostate cancer. Of course, there is biopsy, and there are imaging tools that helps to guide biopsy and also help to detect more advanced forms of the disease. And using these tools together, we can diagnose prostate cancer and define the prognosis. So for localized patients, we can stratify the patients into risk groups, so low, intermediate, and high. And when we talk about localized disease, there are mainly 2 types of treatments with curative intent. One of them is radiotherapy, and the other one is radical prostatectomy, a surgery to remove the prostate gland. And some studies have shown the difference between the outcomes following both methods, and it seems that the outcomes are very similar following 10 years from the treatment. So it is considered to be similarly effective to treat patients. So why some patients should be treated with radiotherapy and not a surgery, or the opposite? So there are some factors that are associated with the disease. For example, older patients or patients with some medical conditions that place them at a higher risk of a surgery will probably go for radiotherapy. But unfortunately, although the two methods are effective to treat patients, there is a risk to develop biochemical recurrence, BCR. And it is defined as the raise in the blood level of PSA after one of the treatments here. The risk to develop the BCR is about 25 to 50%, which is a lot. And unfortunately, BCR is associated with an aggressive disease, more chances for metastases, and higher mortality rates. And as you know, our study focuses on that method, radiotherapy. And after radiotherapy, one of the risk factors to develop BCR is radioresistance. And in this context, we can talk about what is radiotherapy. So for radiotherapy, we usually use mainly three types of radiation methods, which are ionizing, alpha, beta, and gamma rays. Alpha is the usage of protons and also neutrons, beta, electrons, and gamma, photon beams. And these methods differ in their penetrating capacity, and therefore they are appropriate to treat different tumor types and depending on where they are in the body. There are also some modern methods like proton beams that are effective and also cause less damage to normal cells. So they can be used to treat children or near critical area. But overall, all methods are being used, while photon beams is the most common method. And now the question is, how does radiotherapy actually kill cancer cells? And the answer to this question is complex and not exactly fully understood, but we definitely know several things. So first, we know that DNA is one of the major targets that is affected by radiotherapy. Radiotherapy causes ionization, and that can affect the DNA directly or indirectly, and eventually that can lead to the cell death. But DNA is not the only molecule affected by ionization. There are also proteins and lipids in the cell and also other molecules. And finally, radiotherapy can also trigger the immune system to fight against the primary tumor, but also against the tumor metastases under a phenomenon called abscopal effect. Taken together, the effect of radiotherapy on the cell is wide and involves multiple factors. And to add to this complexity, the response to therapy also different between cell types. And of course, that the response also different among patients because there is a wide tumor heterogeneity and different compositions of microenvironments, and that results in different responses to treatments. So here, unfortunately, some individuals will develop radioresistance, which is associated with BCR and an aggressive disease, while some will not. And how can we predict which individual will develop radioresistance? So we don't have enough tools to answer that, unfortunately. But if we did, maybe we could define better which individuals should be treated with radiation and which with surgery. Therefore, in the bottom line, it should be helpful to understand better how radiotherapy affects the cells at the molecular level, and also to identify factors that control radioresistance. And in terms of the different protocols for radiotherapy that are available for patients, so there have been several protocols. One of them is the conventionally fractionated radiotherapy, CF. So CF radiotherapy is a traditional method, and it is characterized by small radiation doses delivered daily over several weeks. And there is an advantage for using small radiation doses if we want to keep the normal cells as safe as possible, because small radiation doses are less toxic compared to high radiation doses. But in the last 2 decades, advanced image guidance technologies have been developed with the goal to increase precision of targeting exactly the tumor site and minimizing the damage to the normal cell. And this picture illustrating the plan for IMRT, one of these methods. And in IMRT, the radiation beams can be conformed exactly to the shape of the tumor. The beams can be delivered from multiple angles and different intensities, so that promise that the tumor will receive the highest intensities and the damage on the normal cells is minimized. And these advanced technologies have encouraged us to use hyperfractionated radiotherapy methods, HF. And HF is relatively new compared to the traditional CF. It is already being used in several places in the world, included in the US. And it is characterized by high radiation doses delivered over only several single treatments. So in terms of the number of treatments, it is much shorter. It is probably more convenient to the patients, of course. And also it is cost-effective. And the fact that we are using here high radiation doses increased potentially the damage that can be caused to the tumor cells. And about the patient outcome following CF compared to HF treatment. So there have been studies that tested that. And in some of the studies, as you can see here, they didn't find a lot of differences in terms of the patient outcome when they tested the biochemical and disease failure, metastases, and mortality. But for some other studies did find that HF is a bit better when testing the disease-free survival, and it is statistically, it is a significant difference. But in any event, radioresistance phenotype can still emerge following both CF and HF treatments. And although the studies that you just saw that tested the patient outcome differences, no one have tested the biomolecular level differences between the cells, so cells that are treated with CF or HF radiotherapy. And therefore, we do not know if similar factors underlie resistance to CF and HF. And it is important to know what are these factors to find the right treatments for patients. And here comes our study to fill this gap. So what we wanted first was to characterize the biomolecular determinants of prostate cancer, of radioresistant prostate cancer. And then we also wanted to test the differences between the radioresistance that is generated after a CF treatment compared to HF. And in collaboration with Dr. Stanley Liu and Dr. Thomas Kisslinger from Toronto University, we were able to model radioresistance using cell lines and to characterize them. So we started with the D145 prostate cancer cell lines, which were originated from human tumors, and they are considered to be relatively radiosensitive. And these cells were treated with both CF and HF according to their treatment protocols until eventually the CF and HF radioresistant cells have been created. Then we took several replicates from each of these cell lines and we tested the differences between them in terms of their genomes, transcriptomes, and proteomes, and also we did some network analysis of genes. And using this cell line investigation, we were able to choose the top targets that we think have the best potential to help for therapy, and we validate the relevance using patient data. And I will start with the results, the cell line investigation. So the first thing that we did was to look on their genomes, and we wanted to test for mutation that gained in either CF or HF radioresistant cells compared to the control. And this plot here shows the number of mutations in each cell. So you can see here that for CF, the traditional method, there are much more mutations compared to HF, the high-dose radiation method, and this difference is statistically significant. And when we tested structural variants, SNVs, so the numbers are lower and also there are no significant differences. So we moved on with the SNV single nucleotide variants, and we wanted to look for mutational signatures that characterize each cell. And I'll just define what mutational signatures are for those who haven't heard about it yet. So that can be defined as imprints left on the cancer genome by a mutational process. And these imprints on the genome are basically the type of the mutation, the type of the base change, the amount of the mutations compared to other, and also where the mutation is located on the DNA in terms of the proximity to other nucleotides. And we can take each profile and associate it with a pool of signatures that exist in the literature. And some of these signatures also associated with some mechanisms. And I think that the simplest way to think about it or to understand that, if taking, for example, tumors from lungs of smokers. So it has been found that people that smoke and have lung tumors, so many times these tumors will be associated with C2A changes. And therefore, this mutational signature was found to be associated with tobacco smoking. So back to the cell line story, we found the mutational signatures that characterize each cell line. And you can see here the composition of mutational signatures. And there is one signature, the orange, that is common to both CF, the traditional, and HF, the high dose, but it has unknown mechanism associated with. But there are also some mutational signatures that are unique for each line, like this purple and yellow for CF and these three for HF. And interestingly, while these mutational signatures were unique to each radioresistant cell lines, somehow they were all associated with defective DNA mismatch repair mechanism. So the conclusion here was that despite differences in numbers and profiles of mutations, overall, these mutations are largely the consequence of defective DNA mismatch repair. We moved on and we wanted to see how these SNVs affect driver genes. And it was important for us to test this because in each tumor, there are many passenger mutations, but there is only a subset of mutation that actually drive cancer evolution and confer the unique characterization of this tumor. And we wanted to see which driver genes drive the evolution of our radioresistant line. And as you can see here, indeed, there were driver genes that were affected strongly, predictedly, by missense variant or splice region variant. And most of these genes are unique to either CF, the traditional, or HF, the hydros, except of this gene that has mutations for both of them. Moving on to the transcriptomics part. So here we wanted to test for differences in RNA abundances of transcripts, and we wanted to focus on cancer-related genes. And therefore, we started with the cancer hallmark pathways and the genes that affect these pathways. And this heat map showed the differences of transcripts. So each row here is a transcript, and each column is a sample. So 3 samples for CF, 3 for HF, the high-dose method, and 2 for the control. And red means high abundance of RNAs and blue low abundance. So you can see here that for CF, these 3 samples, the profiles are pretty distinct from both HF and parental that are largely different and from HF, but similar between them. That is true here, and it's also true here. So we also extended the analysis to test dysregulation of driver genes, now at the level of RNA. And these dotlet present the dysregulated driver genes, here the names, and the type of test that we did. So CF against the control, HF, the high-dose method compared to the control, and both radioresistance lie between them. And the dot size represent how large was the difference between the transcript, and the color is the direction. And every square that is colored with any color that is different from white means that the result was significant. So you can see here that strikingly, almost all hits that we got were in CF cells. So CF has much more dysregulation of driver genes. And when we tested HF compared to the parental, you can see that almost all squares here are blank. So that means that HF also, in terms of driver genes, much more similar to the parental. And if we think about what we have seen so far, so first we saw that CF has more mutations. Second, we saw that at the RNA level, in terms of cancer-related genes, the dysregulation was far more noticeable in CF. And all of this pretty much makes sense, right? Because CF is characterized by small radiation doses. Small radiation doses are less toxic to cells. So that means that the cancer cells have better chances to survive. And if these cancer cells have better chances to survive, they can also gain new mutations. And these mutations can confer advantages, as we can see here. And the fact that also CF has much more rounds of treatment compared to HF also increased the opportunities to the chances to gain these mutations. So for us, it really made sense. And what we can also notice is there are several biomarkers for radioresistance that are known according to the literature that we also identified, like CDH1 and the heat protein 90AA1. And it is always a good control to our study that we also detected them here in CF cells. And now the question can be that if we see all this dysregulation at the RNA level, how much of this dysregulation actually translated into a functional difference at the protein level? And we can see part of the answer in this dot map that shows all the driver genes that were dysregulated at the RNA level, and we could detect at the protein level, so not all of them were detected. And for the proteomic analysis, we were lucky to have the levels of the protein separately for each subcellular fraction. So we have the cytosol, microsome, and nuclear pellet, and so on. And we can see here that 9 out of all driver genes that we detected were also dysregulated at the protein level. That makes it about 40%. And what you can also notice that some driver genes were dysregulated in whole cells, but mostly they were dysregulated in specific subcellular functions, like here, these driver genes that were dysregulated in the nuclear pellet of CF cells. Here in the microsome, in some of the cases, these drivers were dysregulated in both CF and HF. And more in the proteomics analysis, so we also did WGCNA analysis. So we took all these subcellular fractions, and using the software, we could detect consensus modules, which defined as groups of co-expressed genes in both CF and HF data sets. So each color here represents this module of genes, group of genes, and the color showed the difference between the enrichment of these modules in CF compared to HF. So blue, enrichment in CF compared to HF, and red in HF compared to CF. And we can definitely learn a lot from this figure, but I only highlight one of these. So we can see that in the nuclear pellets, there are 3 modules that are colored in blue. So that means they were enriched in CF cells compared to HF. And we took these modules and also did enrichment for biological processes. So we can see here the top 2 hits for each module. And for one of the modules that is colored here in light blue, we saw that the top 1 association was the DNA repair. So that means that there is high activity of DNA repair in the nuclear pellet of CF cells, and that makes sense that the DNA repair happens in the nuclear pellet. And when we look at the same module in the cytosol, so we can see that the color is red. So what red means that the proteins are more abundant in HF, in the cytosol. So what we might learn from that is that since the proteins are translated mostly in the cytosol, maybe in HF cells, these proteins just do not migrate to the nuclear pellet to be activated, but it does happen in CF cells. So that learns something about the differences between the processes in these cells. So that was the cell line investigation. And based on this part, we were able to detect the list of top targets that we wanted to assess to see that they have also clinical relevance. So we used patient data from the CPC gene cohort. It included 382 individuals. Some of them were treated with radiotherapy, some of them with a surgery. And the first thing that we did was to take the list of these top targets that included, for example, driver genes that were affected at the RNA level, protein, or both. And we tested if these patients that we have gain mutation in these target genes. And then we tested if there is a differences in the frequency of this mutation between patients that were treated with radiotherapy compared to surgery. And as you can see here, all genes that are listed here were affected. So that means that the red dots mean that there is a higher frequency of mutations in these driver genes in people that were treated with radiotherapy compared to surgery. So that means that in patients that are treated with radiotherapy, they have higher risk to have mutations in the top target genes that we found. Then we also wanted to test if these genes associated with biochemical recurrence. And indeed, some of the top targets in our list were associated. All of these are significant. We tested here the CNA copy number aberration, so that can be amplification or deletion. And you can notice that the hazard ratios are larger than 1 in all cases. So that means that the risk to develop BCR is increased if there is a mutation in these top targets that we identified. So some of the target genes are prognostic. And also, some of the top targets had mutations that their frequency was first higher in patients that were treated with radiotherapy, and also they were associated with BCR. So we consider these targets as those with the highest level of clinical interest. And with that, I'll summarize. So we first saw that collectively, we observed a far more aggressive phenotype in CF cells compared to HF. Second, we could see that using our study, we can provide a platform for the development of therapies for radiorecurrent prostate cancer, depending on the treatment schedule. And then we demonstrate how ongoing proteogenomic integration can help us to characterize radioresistance. And I want to thank to all people that have contributed to this work listed here. And this work was co-supervised by first my mentor, Paul Boutros from UCLA, and Thomas Kisslinger from Toronto University, who did the proteomics and the subcellular fractionation and many more. and also Dr. Stanley Liu, who have created the radioresistant cell lines, and now together with his grad student Gavin Frame, they are working on very exciting experimental validation for the targets that we identified. And of course, I also want to thank to our group, the Boutros Lab members, and the funding. So I'm funded by EMBO Postdoctoral Fellowship and also the Prostate Cancer Foundation Young Investigator Award. And this work was enabled thanks to this grant lister here from the NIH and the Prostate Cancer Foundation. And I'm happy to take any questions.